Good morning, guys. So it's about 10 o'clock, so I'll make a start. This morning's session is going to be on the nervous system, and this session is going to run from 10 till 2. Okay, so my name's Caitlin. I'm a third-year medical student at the University of Sheffield, and yeah, I'm going to be teaching you today. So if you've not been to an ACE Medic session before, so it works. I'm going to go through some of the content with you. We're going to do a few exam questions and a bit of practice, and then there'll hopefully be a little quiz at the end if we have time for it as well. So some of the things that we're going to cover today, we're going to cover the roles of sensory receptors, the structure and function of neurons, action potentials, transmission, and synapses. And then here's a little timetable of roughly sort of how the sessions are going to end. We might alter things around a little bit if timings change and whatnot, but yeah, so this is just a rough outline of what's going to happen. Okay, so we'll get straight into it. So first of all, I'm just going to start with doing a quick recap of what you learned at GCSE about the nervous system, just to sort of get you introduced into things. So first of all, what exactly is the nervous system? So the nervous system allows us to coordinate our behaviour and respond to our surroundings. And what does that mean? So that means that, so for example, if I accidentally touched a hot pan and burnt myself, signals would be sent through my nervous system and cause me to pull my hand away and to stop it from getting burnt. So this image here shows a little diagram of how that happens, and we're going to have a little look later on today in more depth as to how reflexes happen and to how sensory receptors work. So here we have our stimulus, which in this case is cactus. We have our receptor, which is in our finger. We have a sensory neuron, which takes it to the brain or the spinal cord. We have a relay neuron, which we'll learn more about, which sends the signals around the brain or spinal cord. And then we have a motor neuron, which is going to our effector. And our effector in this case, if we burn ourselves on a hot pan or if we touched a spiky cactus like it is in this image, our effector is our muscle. And the signal causes the muscle to contract and causes our hand to jerk away. So it stops us from getting hurt and stops us from danger, essentially. So our nervous system is made up of lots of nerve cells called neurons, which you might have heard of. So neurons essentially act a little bit like wires. They're sort of the biological wires within our body, and they transmit electricity from one point to another. And so we can split our nervous system up into two main parts. We have something which we call the central nervous system. You might see this shortened to CNS when you see it written around. And it's just a shorter way of saying the brain and the spinal cord. So the brain and the spinal cord are called our coordinators, and they sort of decide what we're going to do about the stimulus, pretty much. And then we have the peripheral nervous system. You might see this abbreviated to PNS for peripheral nervous system. And the peripheral nervous system essentially refers to everything that isn't the central nervous system. So our peripheral nervous system are all of the neurons, so all of those wires extending out from the CNS, so from the brain and the spinal cord, out into the limbs and organs within our body. So nerves that are going from, say, our spinal cord all the way down to the tips of our fingers, they are part of the peripheral nervous system. And then there are also gaps between these neurons where they connect together, and these are called synapses. And we're also going to talk a little bit more about those later as well. So at GCSE, you will have also learned about these things called reflexes. So reflexes are things that are rapid and automatic. We don't think about them. It means we can respond to things really quickly, and it stops us from getting hurt. So as humans, we have adapted we have adapted to have reflexes to keep us away from danger, and we know that having reflexes will help with this. So in our example earlier of the hot pan, we have receptors in our thumb, which when we touch the hot pan, these go off and they send a signal down our sensory neuron. This signal is coordinated, if you like, at the spinal cord in the CNS. And then the response is sent down the motor neuron. And the response in the case of a hot pan is muscle contraction. So we pull our hand away from that stimulus. So those are all things that you should be familiar with from GCSE, and just gives you a little bit of a flavour as to what's going on in the nervous system. So the first thing we're going to start by talking about this morning, which is new to A-level, is a sensory receptor. So what exactly is a sensory receptor? What do we mean by that? So a sensory receptor is nerve endings that respond to a stimulus. And this stimulus can be in the internal or external environment of an organism and can create action potentials, which we'll also learn more about later. What do I mean by the internal or external environment? So for example, our internal environment, that's all of the organs that make us up. So say a signal is being sent from our stomach or something like that, these sensory receptors can respond to that signal. But they can also respond to signals in the external environment. An example of that would be a pressure receptor. So if I say touched an object, if I touched something that was hot or something like that. So, and what exactly is the role of these receptors? So the role of these receptors is to change our stimulus into an electrical signal, and which our nervous system can respond to. So initially, the stimulus will be something like, for example, in our eyes, as you can see in this part of the table, we have cells called rods and cones, and they respond to changes in light intensity. And the role of the sensory receptors in the eyes is to change these changes in light intensity, is to change them into electrical signals. And these electrical signals can be understood by our nervous system. And so that allows us to respond to what we're looking at. Another example is like a change in temperature. So for example, if we're particularly cold or particularly hot, this change in temperature is detected by receptors in the skin and the hypothalamus, and this heat energy is changed into electrical energy. So it's that starts inducing all of the responses. So if we're cold, our brain will then tell us to shiver to try and get us to warm up again, and will cause our blood vessels to constrict to try and conserve heat as much as possible. But the ones we're going to be looking at most today are changes in pressure on the skin. We're going to be looking at a specific receptor called a Pacinian corpuscle, and we're going to have a little look at how that works as a sensory receptor and how that tells our brain how to respond to certain stimuli. Okay, so sensory receptors are very specific. So one type of receptor is specific to one kind of stimulus. So for example, those rods and cones we were talking about in our eyes, they are only specific to light intensity. Our eyes can't detect changes in temperature or changes in pressure. Well, the rods and cones can't anyway. So a sensory receptor is specific, and that's something really important to remember. And that's the same with all of our receptors. So our Pacinian corpuscle that we're going to talk about in a moment, that is only specific to changes in pressure. That can't respond to changes in temperature, and it can't respond to changes in light intensity. There are other receptors which do that job. So sensory receptors act as what we call energy transducers. And what we mean by that, a transducer is simply something that converts energy from one form into another, which is what's written down here. And that's what we mean by a transducer. So there are 4 main types that you guys need to know about, and they've got 4 different names. So we have our mechanoreceptors. This is the general term for the receptors which detect changes in pressure and movement. So for example, we have our Pacinian corpuscles. I think there's another example over here, Meissonier's corpuscle, and lots of different pressure receptors, which are all over our body. We then have something called chemoreceptors. Chemoreceptors, kind of as hinted to in the name, chemo is a bit like chemical. So these detect chemicals. And so those are our receptors involved in smell and taste. So they're able to detect sort of any changes in chemicals. Our next group of receptor is our thermoreceptors. Thermo, like a thermometer, these detect changes in temperature. So those are the receptors working when we touch that hot pan. And then finally, we have photoreceptors. So photo, like taking a photograph, these detect changes in light intensity. So an example of those are our rods and cones, which we talked about earlier. But today we're just going to concentrate on Pacinian corpuscles. It's got a bit of a funny name. You probably won't have heard of one before. I know I certainly hadn't before I came to A-level. So what is a Pacinian corpuscle? A Pacinian corpuscle is a type of mechanoreceptor which detects changes in pressure on the skin. So our Pacinian corpuscle looks a little bit like this. They're found predominantly in our skin, mainly in our fingertips and on the soles of our feet as well, which is written down here. So this receptor is found in our skin, and it allows us to generate a signal which goes to the brain and responds to a stimulus. But how does it go about doing this? So here's sort of my kind of basic diagram of a Pacinian corpuscle. So a Pacinian corpuscle is made up of lots of different elements. So here we have, in the middle here, this is our nerve ending. So this is our sensory neuron, just here. And then we have layers of connective tissue. I've only drawn one here, but there are lots of layers of connective tissue, if you can imagine. Okay, so these are the things that are found in the skin. Lots of layers of connective tissue, and in between these layers, we have kind of a gel-like substance. And this gel-like substance just allows our stimulus, so the changes in pressure, to be sent quickly through, to be transmitted quite quickly through our pressure receptor, through our Pacinian corpuscle. So in the center here, as I said, this is our sensory nerve ending, this bit here. And then these little things here, these represent our sodium ion channels. So we'll learn about those in the next slide, but they're really important. When a Pacinian corpuscle isn't stimulated, those are all closed. And these positive charges here are just to represent that there is a positive potential outside of the sensory neuron, and inside of the sensory neuron, we are at a negative charge. So when our Pacinian corpuscle isn't stimulated, so there's no pressure, it's not, so say for example, you're not touching something or anything like that. I think there's been a bit of a mistake with our link. Sorry, I've just found that out. So who's meant to be in year 12 or year 13? I'll just sort that out for the moment. Yeah, so anyone who's in year 12, if you go into the chat box, they've just popped a message in there. Okay, so the year 12 class will start once again when you click on the link. Do I have all the year 13s? Yeah, so this class is for year 13s only. I'm really sorry for the misunderstanding that took place there. Hopefully everyone who's in the class now is yeah, all year 13s. Okay, sorry about that. I'll just keep going with my with the teaching. So Pacinian corpuscles. So when there is no change in the pressure, so when there's no change in the pressure and there's no stimulus. So say, for example, now my Pacinian corpuscles will be working because I'm holding this pen, because there are changes in pressure on my fingertips on the skin. So when our Pacinian corpuscle is at rest, we say that our cell has a resting potential of around minus 65 millivolts. So there is a higher concentration of sodium ions within the connective tissue than inside the sensory neuron ending. So this is what I've represented here by my positive charge. So there is a much higher concentration of sodium ions outside of the sensory neuron ending. So within these layers of connective tissue, which I've sort of tried to draw over here. So there are lots of sodium ions here. Within the sensory neuron itself, there aren't as many sodium ions, and so we say that the charge of this is negative. The charge of this is negative compared to the outside, which is positive. Okay, and why is this? So this is due to the presence of what we call sodium-potassium pumps. Oh, I think is there an issue with the I think there might be an issue with the link again. Apparently the link sent them back to this class. I'm not sure what's going on. There's an issue with the year 12 link. Still. So is everyone in here? Are you guys all year 13s? Can you just pop in the chat? Are you all year 13s, or Yasina, you're a year 12? Okay. I'm not sure what's going on exactly with the links. The lady who organises it has, I think, gone to the other class currently. Okay, I'll just try and sort that out. I'm so sorry about this. We're just trying to sort things out. Okay, we'll just try and sort that out. Sort that out for you. Hopefully they'll send, I think they'll send the link again in the chat so you know where you're going. Okay, so there we go. So there's a so if the year 12s could click on that link that's been pressed in the chat, hopefully that'll send you to the right class. Yeah, just let me know if you don't get to the right place, and we'll hopefully sort it out and send you to the right place. Okay, so sorry for the interruption there, but the resting membrane potential. So the resting membrane potential is maintained by these things called sodium-potassium pumps. within the membrane. So this is what maintains our high concentration of sodium surrounding the sensory neuron in this connective tissue, and then the negative potential within the end of the sensory neuron. So how exactly do these sodium-potassium pumps work? So these sodium-potassium pumps are found all over the place, and you'll find them a lot. You'll come across them quite a lot during your biology course. So when there is no pressure applied, so when the stretch-mediated sodium ion channels are closed, the sodium-potassium pump maintains the resting membrane potential. So this pump essentially shuttles sodium one way and potassium the other way. So 3 sodium ions are pumped out of the cell for every 2 potassium ions pumped into the cell. And so this leaves, so if we have a look at the diagram here. So this pump, as you can see, is this one here, and it is sending, if this is the sensory neuron, so I'll pull that SN for sensory neuron, it is sending 3 sodium ions out of the cell and 2 potassium ions into the cell. So there we end up with a much higher concentration, if I put of sodium within the connective tissue outside of our sensory neuron. And because that means we also end up with more positive charge outside of our sensory neuron. And then within the cell, this is more negatively charged. We have these potassium ions are coming in. However, there's sort of another little caveat to this, is that although 2 potassium ions are pumped into the cell, the membrane is actually much more permeable to potassium ions. So these potassium ions don't really want to be inside the cell, and so these can actually diffuse back out again as well. So these factors mean that allow the membrane potential to remain at around -65 millivolts. I think anywhere, you'll probably see it written anywhere between -60 and -70 millivolts, anywhere around that area. There's, I think, a bit of a grey area there. But these factors, this sodium-potassium pump allows the resting membrane potential to be the same. So that's what happens before there is any stimulus there. That's how it is maintained. What happens when our Pacinian corpuscle is stimulated and a pressure is applied to, say, for example, our fingertips or to the soles of our feet? So the membrane of a Pacinian corpuscle contains these Na+, K+ pumps, but it also contains what I showed you earlier, which are these stretch-mediated sodium ion channels. So these stretch-mediated sodium ion channels are only activated when there is a stimulus, and they're very sensitive to movement. So, for example, I think here we have one here. So when our Pacinian corpuscle is deformed, so if you imagine that this is me touching the pen, this is what's happening. So that big arrow is the pen touching my thumb. Okay, so you've applied pressure to a Pacinian corpuscle, and because these are quite soft and squidgy, it's caused the membrane to deform. So we've now got this indent within the membrane, and this also causes these sodium ion channels, which are here, to change shape. So these are what we call stretch-mediated sodium ion channels. So when we apply pressure to the Pacinian corpuscle, these stretch-mediated sodium ion channels are able to open. And what's going to happen here? So if we think about our rules of facilitated diffusion, we've got lots of Na+ out here. As we've said, we've got this gradient is being maintained by those sodium-potassium pumps. So we've got lots of Na+, a really high concentration within the connective tissue. So when you open these stretch-mediated sodium ion channels, our Na+ is going to go from a high concentration, which is out here, to a low concentration, which is inside the sensory neuron. So it's going to move down its concentration gradient, the exam board love those words, via facilitated diffusion into the end of the sensory neuron. So what happens here? So sodium ions are positively charged. They have one positive charge, and it causes an influx of positive charge into the end of the sensory neuron. And this leads to the membrane being depolarized and a generator potential being formed, which we'll talk about in a moment. So our cell here was once about minus 70 millivolts. Now we've had this influx of positive charge. The inside of this sensory neuron is probably now going to be more like minus 55 as well. So here we've got sort of like further along in the diagram. So we call this a generator potential. So when these sodium ions are going through our stretch mediated sodium ion channels into our free nerve ending, these are generating what we call a generator potential. These are starting that signal, which then gets sent to the brain. So if it's only a very small stimulus, so if you imagine, I'll draw another Pacinian corpuscle. So if you imagine it's a really small stimulus like that, and it only causes there we go. So we've got a really tiny stimulus now in this one here. And what happens is it only causes a few sodium ion channels to open. Maybe causes that one to open. And it doesn't cause any more to open. It's less likely to actually be able to reach this generator potential and to go on to create an action potential. However, if you have a larger stimulus, which is represented by this arrow here, many more of these sodium ion channels are going to open, and much more sodium is going to go inside the sensory neuron. And that's going to tip the voltage inside of our cell from minus 70 millivolts to about minus 55. And at this potential, we can generate something called an action potential, which we'll look at more later. And the action potential is sent along our sensory neurons back to our CNS. So the really important thing to remember is if we have a small stimulus, it might not open enough sodium ion channels. So we don't have enough of an influx of that positive charge. Whereas if we have a larger stimulus, so for example, you press harder on your finger, as opposed to kind of like a light touch on your finger, it's more likely to open more sodium channels. And so it's more likely to generate an action potential. Okay, so here's a quick question to get you thinking about what we're going to move on to next. I'm going to just launch a poll. So hopefully you guys can see that on your screens. But where do sensory neurons carry signals to and from? Is it A, from the receptor to an effector, B, from the CNS to an effector, C, from a relay neuron to the CNS, or D, from the receptor to the CNS? Whereabouts do our sensory neurons work? We sort of briefly touched upon it, but we're going to talk about it much more in a moment. Any more ideas? Just looking for one more answer. Perfect. Well done, that's the right answer. Yeah, our right answer is D. So our sensory neurons carry signals from our sensory receptor, so from our Pacinian corpuscle, to the CNS. And our central nervous system, if you remember, is our brain and spinal cord. And then we've got another question about what we've just been doing. I will relaunch the poll. So true or false, a Pacinian corpuscle is a mechanoreceptor. So is that A, true, or B, false? What do you guys think? If you're struggling, just look at the word and try and sort of dissect it up. So mechano, does that remind you of something mechanical? Mechanical things move. So what do you think? Is a Pacinian corpuscle a mechanoreceptor? Yep, so both answered and both got that one correct. Well done. So a Pacinian corpuscle is indeed a mechanoreceptor. So that is in fact true. Perfect. So now we're just going to move on. Now we've learned about our sensory neurons, our sensory receptors, sorry. We're now going to learn about how this signal from the sensory neuron gets sent up to the brain and gets sent then from the brain back down to the muscle that's going to cause the action. So I touched on earlier, we have these things which are a bit like wires. So we have our neurons. So there are three main types that we're going to learn about. We have our sensory neurons, we have our relay neurons, and our motor neurons as well. So once a stimulus has been detected, the impulse must be transmitted to another part of the body. So once I have pressed on my pen, like I'm doing so now, the stimulus is being sent down the Pacinian corpuscle. And how does that get to other parts of the body? Well, it's got to travel along these neurons. The impulse is carried as something called an action potential, which we'll learn how they work after the break. And this signal is carried along, firstly, a sensory neuron, which you got right from the question there. So a sensory neuron carries signals from our sensory receptor, our Pacinian corpuscle, to the brain and spinal cord. And then we have our relay neurons. Relay neurons are the ones in the middle. These are the sort of tiny neurons, and these carry them from the in between different parts of the CNS. So those ones are almost kind of bridging the gap, if you like. And then our other, those are only found within the central nervous system. And then we have another lot of neurons, which are called motor neurons. And these motor neurons carry signals from the CNS, so the brain and spinal cord, to the effector. And the effector, what we mean by that is that can be a muscle. That can be a muscle, that could be a gland within the body, or amongst many other things. And that allows us to complete our response. Okay, so first of all, we're going to start by having a little bit of a look at the structure of a motor neuron. So a motor neuron sends signals from the CNS down to our effector muscle. And it looks a little bit like this. So our impulse is traveling, as this arrow shows at the bottom, in this direction. So it's going from this part of the cell to this part of the cell. So this is all one big cell. I think that was something I struggled to understand, was that it is in fact all one big cell, and these can be up to 1 meter long, which is absolutely huge when you think about it. And so how do we make up the structure? So we have a little look here. I think the other diagram's probably easier to see. So at one end, we have our cell body. Our cell body contains the nucleus for our cell, and it contains these things called dendrites. These dendrites branch off into the CNS and connect to relay neurons and other neurons, and sort of which the signal then transmits down. We then have this big long connecting wire in the middle, if you like, and this is called our axon. So this is our sort of big long, the sort of main portion of our motor neuron. And along here, this is where our impulse will travel. So our impulse is traveling along this big long thing. And then we have our axon terminal. Now our axon terminal is what connects to our muscles. So this connects to our muscle in the hot pan, our muscle contracts, so we move our hand away from the hot pan. And so these are what connects to our muscle and what causes that effect to happen. And then we have, you might also notice we have this thing called the myelin sheath. The myelin sheath is essentially like a layer of insulation along our wire, which we'll talk more about later. So, as I've just mentioned, our dendrites, which are connected to our cell body, receive impulses from the relay neurons and from the CNS. So those are those bits here. These impulses then travel through the cell body. So they're going through our cell body from our dendrite and travel along the axon. The axon is our big long wire. So we travel along this wire, and then along this wire there may or may not be a myelin sheath. And our myelin sheath is our insulation surrounding our wire. And then between these bits of myelin, we have nodes of Ranvier. So it's not all one big piece of myelin, it's in fact little individual segments along it. And then we obviously have our axon terminal, which connects to our muscle. So how are neurons specialized to their function? So they're really long, as I mentioned, they can be up to 1 meter long. So they're absolutely huge. So they're able to transmit the action potential over a very long distance. If you imagine, if they were much shorter, it would take quite a lot longer because you would have to keep jumping between each neuron, which obviously is a process which takes time. So if they were shorter, the signals wouldn't travel as quickly. And that's less advantageous for us because if you imagine you're in a dangerous situation, we need to act quickly. And if our signals aren't transmitting quick enough along our nerves, then we're going to put ourselves in a lot more danger. So that's sort of a survival instinct we've gained. How else are they adapted? So the cell surface membrane has many gated ion channels which control the entry and exit of ions, which is very important in terms of our action potentials. So for example, our stretch-mediated sodium ion channels. It also contains lots of our sodium-potassium pumps, so we're able to maintain our cell potential. And they which maintains a potential difference across the cell membrane. And that's really important for us to be able to transmit our action potentials. How else are they specialized? Well, they've got lots of mitochondria and ribosomes. You will probably have heard of these things. Mitochondria obviously are the point of respiration within a cell, which is really important when you have cells which are really active like neurons. And ribosomes are always busy creating proteins. And so that's also really important. So there are many dendrites which can connect to other neurons. So we've got lots of these things. So we can connect to lots of different cells at once. And the axon is insulated by a fatty layer called the myelin sheath, which helps to speed up the transmission of neurons. Okay, so that's how they're specialized. Now, how does a sensory neuron differ to this? So here we have our sensory neuron. Our sensory neuron looks a little bit different, you'll notice. So we have our cell body here, which is in the middle. We've still got the axon, like we had before, but our cell body's in the middle of the cell, as opposed to at the end of the cell. And this big long thing here is called a dendron. So don't get confused. So this isn't the axon. The axon is after the cell body. The axon is always after the cell body. The role of the axon is to carry impulses away from the cell body towards the axon terminals. So this is simply an extension of these dendrites, and it's called a dendron. So dendrites are what connects to, in terms of our sensory neuron, remember we're going from our receptors to the relay neurons or the CNS. So our sensory receptor is down here, our Pacinian corpuscle. The dendrites are connected to the sensory receptors, and the dendron connects the sensory receptor to the cell body. So this is our dendron, and we're connected to our cell body, which is up here. And then our cell body is connected to our synaptic endings by this axon. So the axon transmits signals away from the cell body towards the synaptic endings up there. So as you can see, a motor neuron and a sensory neuron are relatively similar, but there are some subtle differences. You've got to remember that the cell body isn't necessarily at the end, is usually in the middle of the cell. And these can also be myelinated or unmyelinated, as you can see by our blocks of myelin sheath. How about a relay neuron? So relay neurons, these are found within the CNS, and these look quite different. So these are a lot shorter because they don't need to travel as far. These are simply just within our brain or within our spinal cord, and they're just sending signals around there. So in a similar way to the sensory motor neuron, our dendrites receive impulses from either a sensory neuron. The impulse is transmitted down the cell this way through the cell body to the axons, which are, as you can see, so much shorter than our sensory and motor neurons. And then we have our synaptic endings, which are connecting to our motor neurons or other parts of the CNS. So this is our relay neuron. I wouldn't worry too much about it, but it's really important to just remember that it's a lot shorter. And also, if you notice, we don't have any myelin. Relay neurons are not myelinated because they travel such short distances and they're within the central nervous system. These do not need to be myelinated. So here you can see the three diagrams and just comparing all of the different cells. So we have our sensory neuron, our motor, and our relay neuron. And it's important that you're able to tell the difference between these. So this is just a test to see what you already know, but which cells produce myelin in the peripheral nervous system? So you might not know this already. I'm just a little bit of a test before we move on and talk about it. So is it Schwann cells? Is it oligodendrocytes? Is it Pacinian cells? Or is it nose of Ranvier? Which one of these four things produces myelin within the peripheral nervous system? The emphasis there is on peripheral nervous system because there are other things for the central nervous system. Okay, so we've got a split vote. We have one person going for Schwann cells and one person going for oligodendrocytes. So that's really good. Those are the two I expected the answers to be for, and I put one of them in there to catch you out. So in the within the peripheral nervous system, our Schwann cells are actually what creates the myelin. Our Schwann cells are attached to a group of neurons, and they produce the myelin and wrap it around the axons of the sensory or motor neurons. Oligodendrocytes are actually found in the central nervous system, not the peripheral nervous system. And then the other two are just there to trick you out, really. Okay, so, and we've got another question. Relay neurons are always myelinated, true or false? I'll just relaunch the poll. There we go. So true or false, relay neurons are always myelinated. Okay, one more answer. Perfect. Yes, so that is in fact false. So our relay neurons are the ones that are traveling really short distances within the central nervous system. So those are not myelinated, and the answer to this question is false. Okay, so I've just produced a little table for you guys to summarize the differences between sensory, motor, and relay neurons. Here, this is what we've been talking about, and it just sort of summarizes it. It's good for you to look back on, so you'll get these PowerPoint slides in a bit, and it's just a nice little table. So I keep mentioning myelin. So what exactly is a myelin sheath? So a myelin sheath is a fatty layer which insulates the neuron. So as we mentioned, it's in these blocks. So we have, here's our axon, we've got our cell body at this end with our make it look a little bit like a flower with our dendrites sort of coming off. And then we've got our axon terminal at the other end. And our myelin sheath is these blocks. It comes in blocks of myelin. So it's not wrapped around the entire length of the axon. It is just simply wrapped around different blocks, which is really important for later. So our myelin sheath insulates the neuron. It acts just like a blanket, just like you would wrap up warm on a cold day. That's exactly what it does. Its job is to increase the speed of transmission along the neurons. So just like a coat or a scarf is used to keep our heat energy in, to keep us warm, the exact same thing happens with the myelin sheath. The myelin sheath is just like a coat wrapped around the axon, and it allows the electrical energy to stay within the axon and to be transmitted along, and less is transferred to the outside environment as well. So a third of all peripheral neurons are myelinated, and all neurons in the central nervous system are non-myelinated. And we'll learn about a myelin sheath actually, as well as being a good insulator. Also, it also changes the way that action potentials are conducted along our cells, but we'll talk about that later. So in the peripheral nervous system, our myelin is made by Schwann cells, which we just talked about. And every 1 to 3 millimeters, there are gaps in our myelin sheath. And these are called nodes of Ranvier. And the nodes of Ranvier will become very important, but that's just the name simply for these gaps here between these blocks. In myelinated axons, our action potentials, which are our electrical signals which are moving along, in fact, they don't travel straight down the axon like this. They sort of jump like this between each of the nodes of Ranvier. And that allows our action potential to travel faster because it's not having to travel the whole length, it's only sort of jumping between the nodes. And also, the myelin sheath is tightly wrapped, so it prevents the movement of ions across the membrane. And so depolarization only occurs at the nodes. So the myelin sheath is really tightly wound around here, so no sodium ions can diffuse inside, into, or out of the cell when you're wrapped with myelin sheath. So it means that depolarization can only happen in these nodes of Ranvier. Non-myelinated neurons are also associated with Schwann cells. However, the Schwann cells are not wrapped as tightly, not wrapped as closely to the neuron. So non-myelinated neurons, action potentials travel as a wave, and they do not jump between the nodes. Okay, so what are some of the advantages of myelination? Myelinated axons can transmit an action potential much more quickly than a non-myelinated neuron. They enable a much quicker response to a stimulus, as the myelinated neurons are sensory and motor neurons. As the myelinated neurons are sensory and motor neurons, which are traveling long distances. So it's really important. The sensory and the motor neurons are the ones that are myelinated, and those are the ones that can be up to a meter long. So they're the ones carrying the impulses further. Non-myelinated neurons are usually found within the CNS, and they travel much shorter distances, and therefore the speed of transmission is not really as important. It doesn't matter so much. So yeah. So now I'm aware that it's coming up to 10 to 11. So I'm aware that you guys probably need a bit of a break. But in your break, hopefully you can be having a little think about this question. This question is 4 marks. So that should give you an idea of how many points you need to make. But if you can outline the ways in which the structures of a sensory and a motor neuron are similar. Okay, so we're looking for 4 marks worth of answer. And if you guys could just have a go at that, and we'll have a look at the answer when we come back from the break. But yeah, so I'm just going to give you a break until probably about 11 o'clock, should we say, or should we say 11:05? I'll give you a 15-minute break. I'll write it down up here. So if we have a break until 11:05, And in the meantime, you guys can be having a think about this question as well. So yeah, I'm sorry about the confusion earlier, but hopefully things are all sorted now. Yeah, I'll see you at 11:05. Okay, so it's 11:05. So we'll make a start again. Hopefully you guys found the question okay and managed to answer it, all right? I'll pop up the mark scheme now. So here we have the mark scheme. It was 4 marks and then a kind of or 3 marks and then a fourth one for it's one of those questions which gets marked for like spelling and things like that. But some of the answers you could have put so we're thinking about outline the ways in which the structures of a sensory neuron and a motor neuron are similar. So in kind of like very simply, they were just looking for dendrites. So they both have dendrites. They both have an axon. They both have a cell body. And that cell body contains a nucleus. Or it might contain mitochondria or ribosomes, which is what it's looking for in the named organelle. And they can both contain a myelin sheath. So yeah, and it says here you can credit saying may have. So it might not contain a myelin sheath because we know that some of the cells in the peripheral nervous system aren't myelinated, but it might do. And also they both contain the voltage gated ion channels as well. And a sodium potassium pump. So well done if you've got some of the marks there. Hopefully you will have done. But yeah, I'll leave the mark scheme up for a little bit. And yeah, and I think you get the 4th mark for if you spelt any of these words correctly, I think as well. So yeah, well done. And then here's another question. Hopefully we can answer this one quite quickly. So the table below lists a number of statements about the functions of neurons. Indicate whether each statement refers to a sensory neuron only, that's S, motor neuron only, or both motor and sensory neurons. So we can do this one together and you can pop your answers in the chat because it's just a letter for your answer. So the first one's been done for us. So both sensory and motor neurons have a resting potential of about -70 millivolts. So which one transmits nerve impulses from the CNS? So if you can just pop in the chat, is it sensory neurons, is it motor neurons, or is it both sensory and motor neurons? So nerve impulses, keyword here is from the CNS. Yep, so well done, that is M. So yeah, so that one's M. And then which ones connect to other neurons via synapses? So is it sensory neurons, is it motor, or is it both? So just pop the letter in the chat, but which ones connect to other neurons via synapses? Yep, so that's both. We'll learn a bit more about synapses later, but sensory and motor neurons both have synapses, so well done. And then which ones, sensory or motor neurons, connect to effectors? Is it sensory, is it motor, or is it both? Remember, when we say effectors, we mean things like muscles or glands or something like that. Perfect. So well done, guys. You've got all of those ones correct. So here was that's just me scribbling out the answers there, but it's M, B, and then M. So you've done really well there. Well done. So that's where the break was. So now we're going to move on to talk about an action potential. So we know we've learned about a sensory receptor. So we'll learn about posinion corpuscles. If you imagine this box is our sensory receptor, call that S. Now we want to learn, we've learned sort of about neurons and the structure in which our action potentials travel. But what exactly is an action potential? That's what we want to learn about today. So action potentials are the next step in learning about the nervous system. So an action potential occurs when a neuron sends information down an axon. So in this image here, information is being sent down through the cell body along our axon to the next point. If this is a motor neuron, it might be sending it to an effector, so there might be a muscle here, or it could be sending it to a relay neuron in the CNS if it's a sensory neuron. Okay. So how are these signals transmitted? So an action potential is a brief reversal of the potential across the membrane of a neuron, causing a peak of plus 40 millivolts compared to our resting potential, which we said was around minus 65 or minus 70. So we talked about earlier how the resting potential of a cell is usually around minus 65 millivolts. And there are different factors at play, which we're going to talk a little bit more about again. And that determines that the voltage is at minus 65. However, when a signal is sent along a nerve cell, this voltage gets a little bit disrupted. It changes. So, and we call this depolarization. We call this a wave of depolarization. So when the cell's in its resting state, we say that the cell is polarized. But when it changes to plus 40 millivolts, when an action potential happens, we say that it's depolarized. So the cell goes from being polarized to being depolarized. And we're going to learn how the process in which this happens. So let's talk about the resting potential again. When there's no action potential, when the nerve cell is doing nothing and is completely at rest, there's no signal being sent along it, the cells are said to be in their resting state. Compared to the outside of the cell, the inside of the cell, the neuron, is negatively charged. And remember, we've talked about this before, so we've got imagine this is our cell here, a very basic drawing of a cell, but our inside is negatively charged compared to our outside where it's positively charged. And we call this a potential difference because it's the difference between the positive charge on the outside, on this bit, and then our slightly more negatively charged inside. So this is the potential difference. And in a resting state, the resting state of a neuron, it's around minus 70 millivolts. That's just sort of a number that you've got to remember. And this charge is maintained through pumps and anions. Okay. So anions, yeah. Cool. So this is a little picture of a membrane, and this is everything that's going on in the background to maintain the resting potential. So we have, whereabouts are they? Here we have our sodium potassium pumps, which we talked about earlier. So we've got the 3 sodium ions being pumped from the inside, which is here, I'll put an I actually, inside to the outside of the cell. And then we have our 2 potassium ions, which are being pumped from the outside of the cell to the inside of the cell. So that's one way. Also within the cell membrane, we have some other channels. We have some potassium channels. These might be open, but this channel here allows potassium ions to go from the inside of the cell to the outside of the cell, down its concentration gradient. So the potassium is being pumped into the cell. So there's a high concentration inside the cell, and then there's a low concentration outside. We'll go through these in more detail in a moment. And then we have these other channels. So these are our sodium ion channels, which we saw earlier, which are closed at the moment when it's resting. And then there's another type of potassium ion channels, if you like, which are also closed at the moment. So if we go through it in more detail, here we have our sodium potassium pumps. So this is within the membrane of a cell. This is the inside of the cell, and this is the outside of the cell. So if you imagine, if I relate this back to a neuron, this is your axon of your neuron, and then this is the inside of the neuron, and then this is the outside of the neuron. So if you think about this is how it's relating to our nerve cells, to our neurons. So this is what's going on. We're just looking at a chunk of the nerve cell at the moment. So we have our sodium-potassium pumps maintain the negative resting potential by pumping 3Na plus out of the cell and 2K plus into the cell. You guys know this. So this is explained in this diagram here. Here we have our 3Na plus and our 2K plus going into and outside of the cell. Okay, and then our other caveat, which we talked about earlier, was the cell is a bit more leaky to potassium ions. So they don't actually stay inside of the cell. So this results in an overall negative charge within the cell and a very high concentration of sodium ions outside of the cell. Because if 3 are being pumped out each time, then I've got loads of these now out here. And because all the other sodium ion channels are closed, there is no way that these sodiums can get back through. So unlike the potassium, our sodium ion channels are closed, so our sodium can't get back in. This means that the concentration just keeps on increasing outside of our cell. Yep. Okay, so another way in which it's maintained, which I've just talked about, is the voltage gated sodium ion channels are closed. This means that the only way for sodium to get into or out of the cell is by the sodium potassium pump. The sodium potassium pump pumps them out of the cell, so there's a high concentration of them outside of the cell, and these things here, these are not open, these are closed, just like in this diagram here, so they can't get back in. Okay, so sodium can only re-enter the cell once there's a stimulus, once the action potential has started. So I've got another diagram here. So our sodium potassium pump is pumping potassium into the cell. It's pumping 2K plus into the cell. For every 3Na plus, it pumps out of the cell, like so. But because there is a much greater concentration of potassium now, because we've pumped them all inside the cell, haven't we? So there's a much greater concentration in here. And by the rules of facilitated diffusion, our potassium is going to want to leave the cell because there's a really high concentration inside the cell. So our membrane has potassium ion channels in it, and these aren't closed where a resting potential. Some of them are open, not all of them, but some of them. So our potassium can leave the cell down its concentration gradient, so it's going from a high concentration to a low concentration of potassium, and it can leave the cell. So now, both of these factors combined, the fact that our sodium is being pumped out of the cell and our potassium is leaving the cell, that makes the inside of our cell much more negative compared to the outside. So there's, although we don't specifically have any negatively charged ions, which a lot of people get confused by, compared to the outside, it's much more negative. That's what we mean by potential difference. We don't mean that the inside of the cell is negative. We mean that comparatively, compared to the outside, the cell is a lot more negative, just because all of these positive ions are being pumped outside of the cell and they're all leaving the inside of the cell. Hopefully that makes a little bit of sense. So that's what happens at rest. How is an action potential formed? So an action potential is like our signal, and it gets sent from the cell body down throughout the axon and on towards the next neurons and the next nerve cells. So initially our membrane is polarized. This is because there is a negative charge. It's negative inside of the cell compared to the positive charge outside. It's not actually negative, it's just a potential difference. It's negative. Okay. So when an action potential goes along, what happens is our relatively negative inside, there's an influx of positive charge and it becomes positive briefly. And we call that depolarization. So it's going from polarized when the inside is negative compared to the outside, to and it's being depolarized, so the inside then becomes positive compared to the outside. So, but not every stimulus that comes along. So what starts these stimuli? It's our sensory receptors. So our Pacinian corpuscle, when our stretch mediator sodium channels open, remember that our Na plus flooded in. Na plus are positive charges. These can stimulate action potentials to happen. If action potentials are stimulated by an influx of positive charge, and we know that our sensory receptor will bring in that positive charge into the cell, this means that it's depolarized, and this forms an action potential. But if we mentioned before, if our stimulus isn't large enough, so say for example the pressure pressed against my finger isn't great enough for me to be able to feel it, then it means an action potential might not be generated, and so a signal might not be sent. So let's look at this in steps. So we start at our resting potential. We've got a high concentration of sodium outside of the cell. Okay. Then our stimulus comes along. This stimulus causes a couple of our voltage-gated sodium ion channels to open. Sorry. Right? And so it allows a few sodium ions to come in. But as you can see, not all of our sodium ion channels are open yet. So a stimulus comes along, such as a sensory receptor being set off, such as our Pacinian corpuscle being set off. As we saw, that generates a few sodium ions. So these come in to the cell. What's going to happen? So our sodium ions are positively charged. We've got positive charges entering a relatively negatively charged space. So that's going to increase our membrane potential. So our resting potential was minus 70 millivolts. And now it's increasing a little bit because there's more positive charge. So this charge is going to increase. So we then, if we reach the threshold potential of the cell, so if enough sodium ions enter the cell to cause our potential to be minus 55 millivolts now, this then causes these sodium ion channels to open, these ones here. So this is all dependent on the size of your stimulus. If your stimulus isn't big enough, only a couple of sodium ions are going to go through, and it's not going to be enough, and your cell might get to minus 60 millivolts, say. But that's not big enough to then open our voltage-gated sodium ion channels, and not big enough to allow the sodium ions to rush in. And that's the next step. So we also have a look. So that was what was happening. This is what's happening inside the cell. And this is just a graph to show the voltage inside of our cell. So here we are at minus 70, which is our resting potential, which is shown here. So here is our threshold potential. So we've got to get to minus 55. We've got to have enough sodium ions entering the cell to get to minus 55. So here are a few, what is called here as failed initiations. So here is where not enough sodium ions have entered the cell. So it doesn't go on to do this bit. But if we have if the stimulus is big enough, and if enough sodium ions go into the cell, we reach minus 55. At this point, more sodium ion channels open, and so suddenly more sodium floods into the cell. And because we've got more positive charge flooding into the cell, this causes our cell potential to go from minus 55 up to plus 40. And this stage we call depolarization. Because if we remember, initially our cell was polarized because it was negative inside. But now we become depolarized, we're turning it positive. So this step we call depolarization. So what has happened? So if we go back to this diagram here, now what's happened is these channels have opened. like this. So we've got more sodium coming in. So now if we imagine all of these pink dots, all of our sodium ions are now coming into the cell. Yeah, and we are at now plus 40 millivolts. So much positive charge has come in that it's actually turned the inside of our cell positive now. Because there are so many sodium ions in there and our sodium ions are positive. So we're now at plus 40 millivolts. Yeah, and that's because our voltage-gated sodium ion channels are open. Our voltage-gated sodium ion channels are named because they can only open when the voltage has reached minus 55. So they are only able to open when it's at minus 55. Okay, and then we get to the top of this curve. Our sodium ions have rushed in and we're now at plus 40 millivolts. But what happens here? So the inside of our cell is positive. When we get to plus 40, that's another important potential. At plus 40, the sodium ion channels close. So these bits, these sodium ion channels have closed now. Okay. And suddenly a different set of channels open. So when you're at plus 40, it triggers another type of channel to open. And these are called voltage-gated potassium channels. And these voltage-gated potassium channels let the potassium ions outside of the cell. So if we remember, there were lots of potassium ions inside of the cell before when we were maintaining our resting potential. So now the plus 40 millivolts has triggered these channels to open. So the blue circles here are representing potassium ions. When these channels open, because we've got a high concentration of potassium here and a lower concentration of potassium over here, it triggers potassium ions to leave the cell. So our potassium ions leave the cell, and so the inside of the cell therefore becomes less positive. It becomes more negative. Okay, and at this point also our sodium potassium channels also open, and so they're back trying to restore that resting potential, transporting the sodium, which has just gone into the cell, out, and then it's also trying to sort of counteract what the potassium's doing, and it's bringing 2 potassium ions back in as well. So here we have our graph once again. So when at plus 40 millivolts, our voltage-gated potassium channels, K+ channels, open. Okay, and the K+s are pumped out of the cell. And also, this is when our sodium potassium pumps also open again. So they're trying to restore everything back to how it was. So as we can see, we're going from plus 40 and now our potential is dropping. And that's because our potassium ions are leaving the cell. So positive charge is now leaving the cell. So it's now going to become more negative. Okay. And we call this step repolarization because we've depolarized. So we were polarized and we've depolarized it. Now we're going to try and go back to the beginning. So we want to repolarize everything. So this part of the graph is called repolarization. And this is stage 3. Okay, what happens next? Well, you'll notice that on our graph here, we have it goes below our resting potential. So our resting potential is minus 70, but it goes all the way down here, probably to about minus 80 millivolts. Well, why does that happen? So our potassium channels take a little while to shut. So there's a delay in the closing of the potassium channels, meaning that too much positive charge has left the cell. So everything is going to become a little bit more negative than it needs to be, more negative than its resting potential. And we call this stage hyperpolarization. And that's just because our potassium ions, too many of them have left. And hyper usually means like too much of something. So think of it like that. So too many potassium ions have left. Okay. And so it causes this hyperpolarization. It causes things to become more negative than they should be. So we now need to, this is step 4, but we now need to return everything back to normal. We want a high concentration of sodium outside of the cell and a high concentration of potassium inside of the cell. So how do we do that? Well, it's just these Na plus K plus pumps working again, these sodium potassium channels. So what they're doing is they're pumping the sodium that was inside the cell back out, and they're pumping the potassium that was outside of the cell back in. And once they've sort of sorted everything out and the ions are back in their correct places, we can once again remain at minus 70 millivolts, which was where we started. Okay. Does that make sense? Can you let me know in the chat if what I've said so far makes sense, or if you want me to go over it again, because I realise it's quite a complicated topic to understand. Yeah, cool. Perfect. So I'll move on. I'll move on to talking a bit now about refractory periods. So what is a refractory period? So at the end of our action potential, as we've just said, everything's in the wrong place. So if you imagine we're here on the graph, our sodium ions are inside the cell and our potassiums are outside, and we don't want that. Everything's in the wrong place. And it takes time for things to be restored back to normal. And we call this our refractory period. So our refractory period is the period in which another action potential cannot occur. Because if you think about it, in our nerve cell, there are action potentials going through them all the time. They're always being stimulated, but there's a certain length of time in between each action potential that the process can't be triggered off again. So we have, these are two different periods. We have our absolute refractory period and then our relative refractory period, which is here in yellow. So our absolute refractory period is when absolutely no more action potentials can be generated because, well, there's an action potential already happening. If we look here, our sodium ions are already flooding into the cell. So in that time, no, another action potential can't be formed. Okay, and then we have our relative refractory period. Our relative refractory period is when there isn't an action potential happening at the moment, but our cell is just restoring itself back to a normal state. So this is the period in which an action potential, it can be generated, but you must, well, it is the period in which an action potential shouldn't be generated really, but it can be, I think, in certain circumstances, but for the purpose of this, an action potential also can't be generated within the relative refractory period either, because that just represents when our cell is returning itself back to normal. So this is what things look like in the cell. So we've got all this, we've got lots of sodium ions. I'll just draw some more dots. Lots of sodium ions in here, and they're busy being pumped out by our sodium potassium pumps, which are over here. And then our K plus is busy being pumped back into the cell through this. Our voltage gated sodium ion channels are closed, as you can see here, and it is just these pumps doing all the work. So here's a quick question, just to check you've been listening. I'll just reset the poll. Okay, at what membrane potential do voltage gated sodium ion channels close? So is it minus 70, minus 55, plus 40, or plus 60? At what potential do the voltage gated sodium ion channels close? We're just going for one more answer. Perfect. We all got that right. So when when the potential of our cell gets to +40, the voltage gated sodium ion channels close and our potassium channels open to restore it back to normal. So this one's a bit more wordy. I'll relaunch the poll. What is hyperpolarization? I'll let you guys read them yourself. Which one of these describes hyperpolarization? Yep, got one answer in. Perfect, got both answers in, well done. So yeah, both got it correct there, but the answer is A. Hyperpolarization is when the membrane potential becomes more negative than the resting potential. So it's because those potassium channels are slow to close, and so too many leave the cell and it becomes more negative, and then it has to be restored back to normal. But yeah, well done, guys. So now I've got a few practice questions on this topic. So let me think, how does this question work? Okay, so figure 1 shows an action potential. Which row in the table correctly identifies the stages of the action potential? So if you guys can, actually you can answer this one via the poll because it's A, B, C, D. If I open the poll again. Okay, so which row in the table correctly identifies the stages of the action potential? So the numbers, the letters in the table are corresponding to these letters on the diagram. Perfect, and you both got that right again, well done. So yeah, so this is just the diagram that I was showing you earlier. So we're starting at our resting potential, which this diagram here is shown as -80 millivolts, but I think we usually say it's between like -65 and -70. Then our cell becomes depolarized, so our sodium ions flood in, gets up to +40 millivolts, and our cell is repolarized, so it goes back to normal, which is 3. And then we have our hyperpolarization, which is where the potassium ion channels are slow to close, and so it causes the membrane to be more negative than it was before. So yeah, well done, guys. Hopefully you've got those steps down. It's really important to remember them. So we've just worked out how an action potential is formed and how this starts, but how exactly are these action potentials traveled? How do they travel along a neuron? Because as I said before, we've just looked at a snapshot. So this is the axon of our neuron again. Here we've got our cell body and our dendrite, our axon terminal down here. So we have just looked at a snapshot of what's happening in here. But how, if an action potential is generated here, how does that actually cause, how does that actually travel along a neuron? So we've learned about one isolated event. How does that travel along a neuron? So we're just looking at a cell. This is our cell here. Here we've got the outside of the cell, and then here's the inside. So if you can imagine, so this is where our action potential is happening. So our sodium ion channel here has opened, and sodium ions are flooding from the outside to the inside of the cell. Our neuron isn't in one, it's not separate blocks. The myelin is, but it's one big long axon. So it's all connected together. So if you imagine, if the sodium ion concentration increases at this end of the neuron, well, a few of them are going to move along and cause depolarization a bit further along, which is shown in this one here. So that's what we're going to talk about now. I'm going to go into more detail about it now. So this was something I think that I struggled to understand, that the neuron is one big long cell. This axon is all one big long cell. It's not made up of separate cells, it's just one big long one. So it's all connected. So the ions can move around freely within it. So it's just like a wire. And for electrical signals to be transmitted along it, a current must be formed. So, and we call these local currents. So here we have, once again, another badly drawn diagram by me, but it illustrates what's going on. So we've got a high concentration of sodium outside of our cell. So this is the inside of the neuron, inside, and this is the outside of the neuron. We've got a high concentration of sodium out here. And as we know, during our action potential, the sodium floods in through our voltage-gated sodium channels, which are here. Our sodium is moving from a higher concentration outside the cell to a lower concentration inside. So we get a kind of a buildup of sodium ions in here. Now, along the membrane, the rest of the membrane, we have a bunch more of these voltage-gated sodium ion channels, but these ones are all closed at the moment because they haven't been stimulated to open yet. Okay. So what actually happens is when these sodium ions diffuse into the cell through facilitated diffusion, we get a buildup of a high concentration over here. But we know from our principles of facilitated diffusion that things prefer to be in a lower concentration. They like to get away from each other. So our sodium ions are going to try to go from a high concentration to a low concentration. So if you imagine at this end of the cell, once we've had a bunch of sodium ions influx in, there's a high concentration of Na+. But over here, there's a low concentration of Na+. As you can see, there are no red dots over here. There aren't any. Okay. So what's going to happen is our sodium, when it influxes into the cell, is actually going to move this way. So our sodium diffuses sideways along the neuron, away from the region of higher concentration. So it's going away from here, where all that sodium's just come in, and it's going to diffuse along here to try and be with the lower concentration of sodium down this end. And as this happens, it's taking its wave of depolarization. So as the positive charges are moving along here, it's causing these parts of the neuron to become more positively charged. So like this. This is shown here. And what happens then? So as our sodium ions are moving along, this is triggering more of the voltage-gated sodium ion channels to open. Because if you imagine our resting membrane potential was minus 70, but because there's more positive charge flowing through, now the resting membrane is getting to minus 55. And so that's the threshold potential that triggers these channels to open. So these channels are opening, more sodium is flooding in. And so, as you can see, our action potential is traveling through the axon. And this is what happens. We call these local currents. And so when we say that this is a wave of depolarization, it's spreading along. So our sodium ions are diffusing sideways, triggering these channels to open, causing more sodium to move in. And our action potential is moving along the neuron. And this process continues until you reach the end of the neuron. Our sodium ions diffuse from a high concentration over here to a low concentration over here. And more sodium ion channels are opened and more action potentials are formed as this is happening. Okay, So does that make sense? Can you pop in the chat whether that makes sense? Because I think this is one of the concepts that I didn't understand when I did A-level. I didn't really understand how the action potential went from one end of the neuron to the other end. Yeah, cool, perfect. If you have any questions, just pop them in the chat, or if I'm going too quickly, just let me know. But yeah, so that is how our action potential is traveling along the axon of our neuron. Okay, and that's what this diagram here has shown. So you can see the sideways diffusion of our sodium ions going along here and causing these channels then to open and more sodium to flood in. Okay, so now we're going to talk a little bit about how myelin affects this transmission. So first of all, I've just got a question for you. I'll just relaunch the poll. Okay. So at what speed do action potentials travel in myelinated neurons compared to unmyelinated ones? So do they travel at the same speed, whether it's myelinated or unmyelinated? Are they faster in a myelinated neuron, or are they slower in a myelinated neuron? Yep, that's brilliant. So I've got both my answers in now, and you both got it right. So yeah, action potentials travel faster in a myelinated neuron. So the answer was B, and we're going to learn how now. So as we talked about before, the myelin sheath is a layer of fatty material, and this fatty material is really tightly wrapped around the axon. So where we've just seen in our local currents, as the action potential went along, the sodium ions flooded in, and it triggered more channels to open. In an axon, in a myelin sheath, this can't happen. So we have these blocks of myelin with these nodes of Ranvier in between, like so, nodes of Ranvier. Well, our myelin is wrapped so tightly around our axon that these sodium ion channels can't open. So none of this can happen. It can't cause depolarization further along the axon, where the myelin is, because it's wrapped so tightly it just simply can't open. So it prevents the entry of both Na+ and K+ ions into the myelinated portions of the axon. Remember, it's just the myelinated portions. This is where the nodes of Ranvier come in handy, because these are the gaps between the myelinated portions. So this fact means that the action potential is transmitted differently. And this method of conduction we call saltatory conduction. And that's an important name to remember. Okay, so looking at this diagram here, we can see our blocks of myelin sheath, and circled here for you are our nodes of Ranvier. Okay, and we say that in a myelinated axon, our action potentials jump. So they jump from one node to the next node to the next node. Whereas in this diagram here, we see an unmyelinated axon. And in this one, it goes all the way along, like I described before. But in our myelinated axon, it jumps. And it's this jumping that allows it to be quicker. So what happens when the axon is myelinated? At these nodes, an action potential set off. So just what we heard before, our sodium ions are flooding into our axon. So we have that influx. But then, obviously, with our myelin, there aren't any more sodium ion channels along here, or if they are, they can't open. So we don't get the sort of the influx along here. We have to jump to the next node. So we know that where these sodium ions come in, there's a really high concentration of sodium ions here within this neuron. And we know from our laws of diffusion that when at high concentrations, the sodium ions are going to try and diffuse towards areas of low concentration. So our sodium ion, just like it did before, diffuses sideways along the cell. So it diffuses sideways towards the next node. Okay, just like it did with our local current, but at this time, it can't set off any action potentials in between because the myelin is too tightly wrapped here. So it simply just diffuses along. Once the ions, the Na+ ions, have reached the next node, it sets off another action potential. Okay, so when the Na+ has come here, so this is at -70 at the moment, we've not got an action potential. Our sodium ions come along, and because they're positively charged, our voltage is now -55 millivolts. This is the threshold potential for our voltage gated sodium ion channel, which if we imagine is here, to open, and for more sodium ion channels, more sodiums to diffuse in. And so this causes another action potential. So as you can see, we have an action potentials occurring at each node. So our action potentials appear to be jumping from one node to the next node. So we say that our action potentials jump between each of the nodes of Ranvier, just like as shown by these arrows on this diagram. And so because there aren't as many action potentials happening as there would be in this neuron, this actually speeds up our transmission. And so it can conduct an action potential at around 120 meters per second. That's really fast. If you can imagine that humans can run 100 meters in probably, how fast does Usain Bolt run it? Is it 8 or 9 seconds? So this is traveling 120 meters in 1 second. So that's really fast. And these signals are so fast. And that's much faster than it occurs in a non-myelinated neuron. So in a non-myelinated neuron, we just have our local currents, as we call them, which trigger action potentials at all the voltage-gated sodium ion channels along the way. And you can imagine that that process takes a lot longer because there's a lot more going on. Whereas in this one, the sodium ions are diffusing further sideways to get to each node, but there's fewer action potentials happening, so it's much quicker, if you see what I mean. So we also have another concept to sort of talk about, which is all or nothing transmission, which is really important to get your head around. All action potentials are the same intensity. So basically, if a stimulus generates an action potential, it doesn't mean if that stimulus was really hard. So say, for example, was a weight falling on your finger, or whether it was a feather touching your finger, that will generate the same intensity of action potential. Because if an action potential is generated at -55 millivolts, that sets off the same process every single time. Yeah. So when it reaches -50 millivolts, that sets off the sodium ion channels opening. And it doesn't matter whether, as I say, it was a brick that caused that or whether it was a feather, it sets off this action potential regardless. And we call that all or nothing transmission. So your stimulus might be huge, or your stimulus might be really light and tiny, but provided it reaches that -55 threshold potential, it'll set off an action potential regardless, and it's the same intensity. However, some stimuli are so small that it can't reach this threshold potential, and so it doesn't set off an action potential. And that's the sort of the nothing transmission part of it. However, you can produce, one thing that does affect the intensity of an action potential is the frequency. It's not the size of it, it's the frequency, because it will generate more action potentials. If you're constantly stimulating something and saying, I'm constantly doing this, I'm generating more action potentials. I'm not generating a bigger one, I'm generating more. There's more generator potential. Okay, So we're just coming up to sort of lunchtime now. So I'm just going to do, I've got a couple of questions, which I think it'd be nice for you guys to answer. So I'm going to give you, it's a 2-mark question, so I'll give you a couple of minutes to do this. I'll set a timer. Let me, got a timer ready. So multiple sclerosis is a disease that involves damage to myelin sheaths of neurons. Movement in MS sufferers may be jerky or slow. Damage to the myelin sheaths of neurons can lead to problems in controlling the contraction of muscles. So from what we've just talked about, explain one reason why. So if you guys can have a go on your own piece of paper, I'm going to give you a couple of minutes to do this. And then after a couple of minutes, I'll come back and answer your question. We'll go through the answer together. So if you guys can have a go answering this. Okay, so that was my timer just going off. So that's 2 minutes. And hopefully you guys have had a think about it, and don't worry if you're not quite finished. Right, so let's have a look at the mark scheme. So we're suggesting one reason why damage to the myelin sheaths can lead to problems controlling the muscles. So the first marking point you could have, so what it's looking for here is kind of a suitable explanation that's been explained. So because in multiple sclerosis, the myelin sheath has been damaged. So this means that the action potentials are going to travel more slowly or not travel at all. So the first marking point can be action potentials simply just travel more slowly because there isn't as much myelin there, or there's not myelin at all. And so what happens now? It leads to a delay in muscle contraction. So if you can't send a signal, or the signal is being sent more slowly, your muscles are then going to contract much more slowly as well. So that's kind of the effect of it. And that's what happens in people who have multiple sclerosis. So, and then another, you could look at this question from a different angle as well and say that action potentials, because it's not been surrounded by that myelin sheath. Remember I said that the myelin sheath acts as an insulator and it keeps all the energy in within that neuron. Well, if you don't have your myelin sheath there, if you've not got your scarf on, then you're going to lose more heat energy, aren't you? So it also means that action potentials leak to adjacent neurons, and so that can cause another neuron to be set off, and so the wrong muscle fibers contract. So that can cause involuntary movements in people with MS. So I thought it was a really nice question that sort of related things to medicine a little bit. Let's have a look now. Okay, so we've got 2 more questions here. Let's see, are they there? Yeah, so these are the last questions. I'll give you these ones to have a little think about whilst you're having your lunch, and I'll go through the answers after lunch. But it's 12 o'clock now. So if we come back at about 12:30, put it up here, you can have a little think about the questions in the meantime. And yeah, and I'll see you at 12:30. Okay, so it's 12:30. Hope you guys had a nice lunch. So we'll just start by answering this question, which hopefully you've had time to have a little think about. So the question is, scientists have investigated the use of substances called cannabinoids to control problems caused by MS. Cannabinoids are hydrophobic molecules. In the body, they easily pass into neurons. Explain why. So I'll pop the mark scheme up for this one. So, so the answer to this question is because they're hydrophobic, that this is obviously not directly related to what we've been talking about, but you should sort of have an idea from like other biology concepts. But because they're hydrophobic, this means they don't like water, but it also means that they're lipid soluble. So they can pass through the phospholipid bilayer that's surrounding the neurons and things. So that is why they sort of would make good drugs to treat MS. And then the next question was, cannabinoid receptors are found in the presynaptic membrane of neuromuscular junctions. When a cannabinoid binds to its receptor, it closes calcium ion channels. Suggest how cannabinoids could prevent muscle contraction. So, okay, so this one is a little bit difficult. I don't think you can answer this one yet, actually. We can come back to this one after we've done synapses, which will be next. I realise that this question is a little bit difficult for you guys to do, but this one's about synapses and things like that. So yeah, I didn't realise that one was about synapses. So we'll come back to that one in a moment, and we'll talk a little bit about synapses now. So, so what are synapses? Synapses are the junctions between two or more neurons. So this morning we've learnt about our sensory receptors, the neurons which connect that to, say, the CNS, to the brain. And then now we have these little gaps between the neurons. So a signal, an action potential, when a sensory receptor is stimulated, the action potential passes up the neuron, as we've discovered. But when it gets to a junction between neurons, it's got to get across them somehow. And that's where these things called synapses come in. A synapse looks a little bit like this. This is the presynaptic bulb. And they're used to communicate with one another, as it says here. So the synapses form the junctions between two or more neurons. Or more is quite important there. It doesn't just have to be two. Things can be a bit more complicated. And synapses allow neurons to communicate with one another. So our synapse is made up of a presynaptic bulb, which you can see here. So this is on, so say you've got two neurons, one's this line here and one's that line here. The presynaptic bulb and the action potential is traveling in this direction. Presynaptic bulb is on the end of the first neuron, and then they have a receptor, which is on the end, which is on the start of the next neuron. And between them, we have a gap. And this gap is called the synaptic cleft. I think it'll show, is there a better diagram of it here? Yeah, so here's a better diagram explaining that. So this is the end of our neuron. Here we have the receptor, and then here we have the synaptic cleft. So how do we communicate across synapses? Synapses are a little gap in space. So, but action potentials aren't able to bridge the gap between these two neurons. So we've got to do this another way. And the way that it happens is by use of a neurotransmitter substance. So within these little vesicles here, these little circles, we have substances called neurotransmitters. Neurotransmitters are kind of small little dots that are able to diffuse across the synaptic cleft. And these can bind to the receptors on the other side. So here we have the other side, and they can bind to the receptors in the membrane. And that's sort of how synapses work. And then once these little neurotransmitters bound with this membrane, it sets off another action potential. So that's what a synapse is. And we're going to go into more detail as to how this process happens. But essentially, a synapse two neurons communicate via a synapse, and the synapse works by sending a neurotransmitter, which is a substance, across this gap. So, and this neurotransmitter substance is able to then generate a new action potential. So the synapses that we're going to be concentrating on today are called cholinergic synapses. And these release a neurotransmitter substance known as acetylcholine. You'll see me abbreviate it to ACH throughout here because obviously acetylcholine is quite a long word. So that's abbreviated to ACH. And the neurotransmitter is those, acetylcholine is just simply the little dots here inside these vesicles. Okay, so here's the picture that I showed you earlier. So just to sort of orientate yourselves, this is the action potential that is coming down, say, for example, our sensory neuron. And this is the action potential. It's coming along as a wave of depolarization. It gets to this point here, and it opens the calcium channels. So the wave of depolarization triggers the opening of these calcium channels. Because there is a much higher concentration of Ca2+ outside of our synapse, the Ca2+ can then diffuse into the presynaptic bulb. So Ca2+ diffusing in. And then this triggers these vesicles to move closer towards the membrane, like so. And these vesicles then can release their neurotransmitter, so the ACH, across the membrane, which is where they bind. I'm going to go through that a bit more slowly now. So, what are the main structures within our presynaptic neuron? So it's really important that you're able to recognize which bits are which. So this whole thing is called the presynaptic bulb, as I've mentioned, and it contains voltage-gated calcium ion channels, which are these bits here. Also has vesicles, which we mentioned, which contain our neurotransmitter. And then it also has mitochondria, which are here. This is a process which requires energy. So it needs lots of mitochondria to be producing ATP, which is producing respiration. And also, we have a lot of smooth endoplasmic reticulum. If you remember back from year 12, when you will have done sort of like the structures inside of cells, our smooth endoplasmic reticulum, the purpose of that is to package things into vesicles. And within the end of in our presynaptic bulb, it packages our neurotransmitter, so these green dots, into these vesicles, which are these little circles which hold it in place. And then what does our postsynaptic neuron look like? So here we have the synaptic cleft, which is this hole in the middle. And here we have the vesicles, which have bound to the membrane and are releasing neurotransmitter across. And they come to the postsynaptic neuron. So on the postsynaptic neuron, we have sodium ion channels, which are here. And we also have receptors, which you can't really see very well in this photograph, but the neurotransmitter comes along and binds like this to these sodium ion channels. And when the receptor is bound, it causes it to open, which is really useful. These receptors are specific to acetylcholine, so these are cholinergic receptors. So only when acetylcholine binds does the sodium ion channel open. So now you've got an idea of how it works. Let's go through it in stages. So our action potential, which I've shortened to be AP here because it's shorter to write out, arrives at the presynaptic bulb. So that's our, imagine we've got our sodium ion channels, got our sodium ions coming in, and it arrives here. This now triggers our voltage-gated calcium ion channels, which are these things here, and these are found within the presynaptic bulb. It triggers them to open. Now, because there is a high concentration of calcium, which is represented here by these purple dots, outside of the cell, when these voltage-gated calcium ion channels open, these calcium ions diffuse into the cell, and they diffuse into the cell via facilitated diffusion because they're going through a channel, like so. And that is the first stage of synaptic transmission. The action potential arrives, and then that triggers voltage-gated calcium ion channels to open. Okay, and within here you can see also we've got our vesicles, which are waiting and ready for the next stage of synaptic transmission. So what about our next stage? So our calcium ions have diffused into the bulb. So there's now kind of a higher concentration of them in here than there is out there. And they fuse and they cause the vesicles to fuse with the membrane. So these vesicles were here, if you remember, and it causes them to migrate over towards the membrane and then they fuse with it. Okay. So in these photos, they're fused with it. And remember that inside these vesicles, we have ACH. I've got a little key up here if you're ever confused what my different coloured dots mean. And here we have the synaptic cleft. Okay, cool. So now what happens, once these vesicles are fused with our membrane, they are able to release the acetylcholine. So that's these things I've just circled here, these yellow dots. The acetylcholine is then released. And it's released into the synaptic cleft via a process known as exocytosis. So essentially, that's the vesicle sort of just spitting out the acetylcholine. And so the acetylcholine can now cross the synaptic cleft. So yeah, so the acetylcholine molecules diffuse across to these receptors, like so. So that's your next stage of synaptic transmission. So once the acetylcholine has diffused across, it binds to our sodium channels. So here we have our sodium ion channels. And if you remember, there are receptors attached to the sodium ion channels. So if you imagine it, if I do a sort of a zoomed in version over here, so this is a close-up of the sodium ion channel, and then this is a little receptor attached to it. If I put two there, one on each side. And so once these receptors are filled, so this big red blob I'm drawing now is my sodium ion, is my acetylcholine, sorry. Once my acetylcholine binds with its receptor on the sodium ion channel, this allows the sodium ion channel to open. And it allows Na+ to move into the cell. Like so. And got to remember this is called the postsynaptic membrane. Yeah, so if you ever get confused by the names, pre, I suppose, is like before, and post is after. So this is sort of where it is happening before, and then it gets the membrane, the postsynaptic membrane after. So it's pre, then post. So yeah, so then once our sodium ion channels are open, as you can see here, the sodium then diffuses through across the postsynaptic membrane. So it's able to come in. And then if you remember from before, what about our action potentials? Well, our action potentials are started by sodium ions moving into the cell. So that's exactly what's happened here. Our sodium ions have now entered the postsynaptic membrane, and these guys are now able to start a generator potential. And if enough action potentials combine, so you get enough sort of signals going through, enough action potentials coming through, and enough sodium ions, it means that a new action potential can be triggered in the postsynaptic neuron. So just like before, where we have our cell and the sodium ions move in, like so. If we have enough sodium ions come into the cell and they make that generator potential, which we know is minus 55 millivolts, so if they cause the potential to change enough, then a new action potential is started. And this action potential then goes off and down along a new neuron. So that's what happens at a synapse. So hopefully that's broken it down for you into some steps. I think it ended up being 10 steps, but hopefully that's broken down the process for you. Simply what happens is the neurotransmitter, so acetylcholine, diffuses across a gap, attaches to a receptor and sets off a new action potential. So yeah, but what happens to the leftover acetylcholine? So if you imagine we've got our synapse here, which we've just seen, which looks like this, and then our receptor. So a new action potential has been set off, and it's set off going down here. But we've still got acetylcholine bound to these receptors. It's not gone anywhere because we've not done anything with it yet. So these sodium ion channels are still open and sodium ions are still flooding in. And this means that there's going to be kind of a continuous action potential going. So it doesn't stop. But that's not what we want. We don't want our muscles to continually be working, or we don't want signals to be continually sent down on neurons. So there's got to be a way for this to be switched off. So that's where the enzyme acetylcholinesterase comes in handy. So acetylcholinesterase is specific to acetylcholine, hence the name, and it's found in the synaptic cleft. I like to imagine enzymes as little Pac-Man. Let's see. So here's my acetylcholinesterase. So this enzyme is able to break down our neurotransmitter, our ACH, and prevent the transmission of any further signals. So what this does, I'll draw a bigger one over here. Here's our little Pac-Man. Here's our acetylcholine. So what it does is it is able to break this down. So it can no longer stimulate these sodium ion channels to open. So it stops the sodium from traveling through them. And therefore, once the sodium ion channel is closed, it'll stop the action potential from happening. And prevents any further signals until a new action potential comes along down the presynaptic neuron. So what happens to these breakdown products? Once our acetylcholinesterase has broken down our acetylcholine, the breakdown products are then reused and recycled. So these breakdown products are able to re-enter the presynaptic bulb, and they're used to form ACH, or acetylcholine, inside of our vesicles once again. So it's all sort of a recycled process. These materials aren't wasted. They're simply broken down and then used again. Broken down and then built back up again to be used again. But yeah, it's really important to remember acetylcholinesterase because that plays a really important role. If you don't have any acetylcholinesterase, then there's something going wrong, pretty much. And it can cause different kinds of diseases and pathologies and things. So that's how a synapse works. And the exam board love questions about synapses and asking you about what goes on at synapses. But also, we also want to cover a topic called cell signaling. So as we've discussed before, action potentials are very much like all or nothing responses. So when an action potential is generated, when that minus 55 millivolts is reached by the influx of sodium, no matter the size of the stimulus, provided it's reached the minus 55, the same response will happen, and all of the sodium will then start flooding into the cell through the voltage gated sodium ion channels, and so on. However, one action potential in the presynaptic neuron does not produce one action potential in the postsynaptic neuron. So we might need more than one action potential in order to stimulate the postsynaptic neuron, so the one across the synapse from it, to set off. So this is where the whole thing about frequency comes in. If you only have one action potential going along your presynaptic neuron, it's unlikely to generate an action potential within the other one, because you need lots of acetylcholine to stimulate those receptors and lots of sodium to go in and for it to be stimulated. Okay. So you need a series of action potentials in order to reach the threshold potential to create an AP in the postsynaptic membrane. So you need enough action potentials to bring enough sodium and positive charge across into the postsynaptic membrane to be able to set off an action potential in the postsynaptic neuron. So there are different ways of doing this. So, and we call it summation. There are two different types of summation we're going to talk about. So, as I said before, a synapse doesn't necessarily join two neurons. It can join more than two neurons. It can join, so for example, here we have three. We have three presynaptic neurons, and then it's just attached to one postsynaptic neuron. So summation occurs when the effect of several excitatory postsynaptic potentials are added together. So that's when you've got three action potentials, excitatory action potentials arriving at your synapse. This is all added together, and then this allows an action potential to form. So so this is just what I said before. So there can be more than two neurons involved. We can have three on one side and one on the other, like you can here. So you can have, as I've said here, you can have three neurons converging to form one neuron. So like in this example, or you can have it the opposite way around. You can have one neuron converging one neuron and then sending off into three separate neurons. So yeah, you need several action potentials to be able to generate an action potential in the postsynaptic neuron. So when ACH diffuses across the synaptic cleft, this causes a small depolarization, and this is called an EPSP, or an excitatory postsynaptic potential. These are then combined to form an action potential in the postsynaptic neuron. So in this example here, this neuron will create an EPSP. This neuron will create an EPSP, and this one will as well. Combine them all together, and you're able to form an action potential in the postsynaptic neuron. Hopefully that makes sense. It's a bit of a confusing concept to understand. And there are two ways in which this can happen. So the one we've just looked at was spatial summation. So that's the result of three different, well, not necessarily three, of a number of different neurons converging together at one synapse. So you've got signals from lots of different neurons all arriving there at the same time, producing those EPSPs and producing an action potential. Sorry, there's a fly. Then we have another thing called temporal summation. So temporal summation is another way. So in this synapse, we only have one presynaptic neuron and only one postsynaptic neuron. But this time, signals have been sent in sort of rapid signals have been sent. So three signals have been sent really close together. So each of these signals on their own wouldn't generate an action potential, but when they're added together, they do. So this is why the bigger the frequency, the more likely an action potential is to be generated. The action potential will be the same size each time. However, an increased frequency of them will cause an increased likelihood of an action potential being generated in the postsynaptic neuron. So yeah, so just remember that temporal summation is as a result of several signals being sent in the same neuron, whereas spatial summation is as a result of action potentials arriving from several different presynaptic neurons. So you just need to know the difference between those two. Temporal, several signals in one; spatial, more than one neuron. And then just to confuse things even more, we have different types of neurotransmitters. So we just looked at a cholinergic synapse, and that is a synapse which, as we said, uses the neurotransmitter acetylcholine. But you can get different substances which can cross this membrane. It's not just acetylcholine. Acetylcholine is an excitatory neurotransmitter. That means that when it diffuses across, it's going to cause an action potential to form in the postsynaptic neuron, and that's then going to cause something to happen. However, you can have another type of neurotransmitter. Another one I've used an example here is called GABA. GABA is an inhibitory neurotransmitter. This means that it actually prevents an action potential in the postsynaptic neuron. So it can prevent it happening. So here where we have like 3 excitatory signals combining to form an action potential, if instead of 3 excitatory signals, we had 1 inhibitory signal, well, then that would stop the action potential from being able to be formed. It prevents the action potential from forming in the postsynaptic neuron. So yeah, so you just need to know that there are different types of neurotransmitter and they cause different things to happen. You don't need to know any of the specifics about them. But yeah, there are many different types. There are more other than just acetylcholine and GABA as well. So what are the benefits of having this kind of method of controlling our communication between neurons? Well, when several neurons converge into one postsynaptic neuron, this can create a very specific response, which may be beneficial for an organism. So yeah, because it's got signals from 3 different places. So in this one here, you've got signals from 3 different places, and this can be very specific. It's 3 specific places creating a specific response. So that's why, that's one reason why it can be beneficial. If you have an inhibitory signal, like I mentioned with GABA, this can prevent any excitatory signals from being carried out. So if I have 2 excitatory signals and then 1 inhibitory signal, that will then prevent all these signals from being sent. So that potentially could get you out of a dangerous situation or could stop you from getting hurt or something like that. So that's really important. Another way, so one presynaptic neuron can diverge to several postsynaptic neurons. This allows the signal to be passed on to many different areas of the nervous system at once. So imagine you have one signal going on an action potential, and it needs to reach different areas of the brain. So this can be sent from one presynaptic neuron, can then be sent to one area of the brain via this neuron, one area of the brain by this neuron, and one area of the brain via this neuron. So it can converge. So 1 can be turned into 3, and the same signal is passed down those 3 different neurons, and it's letting 3 different areas of the brain know at the same time, or of the nervous system know at the same time, to produce, once again, a very specific response, and quite a quick response as well. So yeah, so those are some important bits. And then there are some other ones as well. So synapses are able to filter out any low-level signals if they don't reach the threshold potential. So say, for example, I'm sitting in a chair currently, and constantly there are going to be constant signals going off the whole time that my legs are sat on this chair currently. So synapses can filter out any of those low-level signals if they don't reach a threshold potential. So they can filter out any unwanted signals, so kind of like a constant stream of signals, it can filter them out because they don't reach the threshold potential. So that's the benefit of having a synapse which needs kind of multiple of the EPSPs to form an action potential. And also on the other end of the spectrum, it can filter out low-level signals, but also low-level action potentials can be amplified. So the process of summation can actually amplify a signal. So if you've got lots of action potentials here going through, if you just had one of them, it wouldn't cause an action potential. Because we've got all three, it does. So in this way, the lots of action potentials can be amplified. And also, just like me sitting in a chair, after repeated stimulation, we may run out of neurotransmitter, and so our synapse is fatigued and our body has become used to it. So there are sort of like lots of different things that our body might become used to over time. And so we're not constantly aware of it all the time, because that would be really tiring if we were constantly aware of sort of everything that we were doing. So yeah, so now I finished sort of the main content for the session. So now I've just got some practice questions and a bit of a quiz to go. So I'm going to go back to that question earlier that we couldn't answer because we haven't quite done it yet. So, okay, here we go. So the question from earlier regarding synapses. So a cannabinoid receptors are found in the presynaptic membrane of neuromuscular junctions. When a cannabinoid binds to its receptor, it closes calcium ion channels. Suggest how cannabinoids could prevent muscle contraction. So I'm just going to give you guys another couple minutes. Now you've got kind of some more knowledge about the situation, to think what you would write for this answer to the question. Okay, so hopefully you've had a little bit of a think about it again. So how can cannabinoids prevent muscle contraction? Let's find the mark scheme. Okay, so the mark scheme's over here on the left-hand side. So we've just learned that in order for a neurotransmitter to cross a synapse, calcium ions flow into the presynaptic bulb. So I'll draw a little diagram here. Calcium ions flow into the presynaptic bulb. We know that cannabinoids receptors are found in the presynaptic membrane, and when a cannabinoid binds to its receptor, it closes the calcium ion channels. So if these calcium ion channels are closed, when an action potential comes to the end of the synapse, the calcium isn't going to be able to diffuse in. And that means that the rest of the steps aren't going to be able to happen. The vesicles won't migrate over towards the synapse, and the neurotransmitter won't be released. So that's sort of the idea that this question's getting at. So if we have a look at the mark scheme over here, so it prevents the influx of calcium ions, so it's really good. Make sure you state that. So synaptic vesicles can't fuse with the membrane, which is the next sort of step, and can't release the neurotransmitter. So therefore, the neurotransmitter doesn't diffuse across the synapse, and it can't bind to these receptors which are on the synapse over here. So no action potential or depolarization of the postsynaptic membrane can happen. So that just completely stops any action potentials from forming. So that's sort of an example of how we can use drugs to cause different things to happen within our neurons. So hopefully that has explained that a little bit. Did everyone find that question okay, having a go at it now now we know more stuff about synapses? Has that explained it a bit better? Just pop in the chat, just yes or no. Yeah, cool. And now we'll do a few more exam questions. Cool. Let me know if you can just pop in the chat whether you're happy with everything that we've gone through so far, or is there anything in particular that you've not understood and want to go back to? If you pop in the chat, just say if you're happy, or write us any specific thing. Yeah, all good. So everything's made sense to you so far. Cool, happy. Perfect. Right, so here's the next question. So synapses are an integral part of the nervous system. Outline the roles of synapses within the nervous system. Okay, so this is a 3-mark question. So I'm going to give you kind of 3 or 4 minutes to answer this question. Have a go, and then we'll go through the answers. We'll go through the answers in a couple of minutes' time. Okay, so that's been 3 minutes. So hopefully you guys have come up with some answers, and we'll go through them now. Okay, so here's our mark scheme. So what was our question? Synapses are an integral part of the nervous system. Outline the roles of synapses in the nervous system. Okay, so there are lots of different things you could put for this question because it's quite broad. So what is the point of synapses? So we know that synapses are our junctions between neurons, as I've just sort of drawn a little diagram up here. So what does this allow? So this allows neurons to communicate, so for cell signaling and things like that. So in the guidance here, it passes impulse onto the next neuron. So it allows action potentials to cross the gap in a way by using neurotransmitters between one neuron and another. It ensures transmission between neurons in one direction only. So you've got to make sure that it's transmission between neurons as well. When you put it here, it's not transmission between a neuron and something else. It's got to be transmission between two neurons. That is sort of the rules for what a synapse is, if you see what I mean. And it's in one direction only because it can't go backwards and work the other way. Another role of a neuron that you might put down allows convergence / impulses from more than one neuron to be passed to a single neuron. So that's what we were talking about, where one neuron can be send out kind of 3 signals to 3 different parts of the brain, or signals from 3 different parts of the brain can converge in one singular neuron, and that sort of thing. So that's what it means here, and that is answers 3 and 4. So that is one of the benefits of transmission via a synapse. Or the idea that it filters out background / low level stimuli, because you need obviously more than one EPSP in order to stimulate an action potential in the next neuron. Which then prevents fatigue and overstimulation. It can allow low level stimuli to be amplified, as we mentioned before. And then the presence of inhibitory and excitatory neurotransmitters allows impulses to follow a specific path as well. Permits memory, learning, and decision making. So there are lots of different answers here which we've talked about. So hopefully, how did you guys find that question? Was that question okay to answer? Do you reckon you got all the marks for that one? If you pop in the chat, just with it was good or not good. Mostly good. Okay, that's good. I think one sort of challenge, obviously, to doing exams is sort of learning what the examiner is looking for and what marks to put in, what points to put in. Okay, so now I've got another question for you here. So serotonin is another type of neurotransmitter, just like our ACH and our GABA. So serotonin is a neurotransmitter released in some synapses in the brain. It is transported back out of the synaptic gap by a transport protein in the presynaptic membrane. Serotonin diffuses across the synaptic gap and binds to a receptor on the postsynaptic membrane. Describe how this causes depolarization of the postsynaptic membrane. Okay, so we're describing how a synapse works pretty much with that one. And then the next question, which I'd also like to have a go at, it is important that a neurotransmitter such as serotonin is transported back out of synapses. Explain why. So I'm going to give you guys 4 minutes to answer those questions. Okay, have a go, and then I'll come back and we'll go through the answers afterwards. Okay, so that was 4 minutes, and we'll just go through the answers now. So for the first question, describe how this depolarization of the describe how this causes depolarization of the postsynaptic membrane. So, serotonin diffuses across the synaptic gap and binds to a receptor. So what we're wanting from this question is, here we have, once again, my presynaptic neuron is getting bigger every time I draw it. So here we have the serotonin. It's just a neurotransmitter, just like ACH, except for it does have a different role, but it's diffusing across. Our serotonin then binds onto the sodium ion channels, like so, and causes the sodium ion channels to open, and causes sodium ions to enter the postsynaptic membrane. So these are the marks here. So when the serotonin binds to the receptor, it causes the sodium ion channels to open. And then sodium ions enter the cell, and this causes depolarization. And if there's enough of them, it'll cause an action potential to form as well. So that was the answer to A. And then B, it is important that a neurotransmitter such as serotonin is transported back out of the synapses. Explain why. So this is the sort of what we were talking about before. So our action potential has been and gone. It's been set off, and we've still got lots of serotonin within our synapse. So it needs to be removed, because otherwise it'll just keep sending signals. It'll keep binding. It'll stay bound to the sodium ion channels, and sodium ions will keep going through. And so it will keep setting off more and more action potentials. So if it's not removed, this will happen. And obviously that's not very nice and causes diseases and things like that. That's the pathology in itself. So yeah, so the marks it's looking for here is, if it's not removed, it keeps binding to the receptors. And it keeps causing action potentials to happen. It also accepts with depolarization here as well. And keeps Na+ channels open as well. So hopefully, how did you guys, can you pop in the chat how you found that question? Was that good, mostly good, bad? Were you able to answer that question okay? Yeah, mostly good. Okay, that's good. Good to hear. So we're just going to have a little break now. We're going to have a little break. Oh dear. We're going to have a little break for about 15 minutes before we come back. I've just noticed there's another exam question as well, which is not done. But yeah, we'll have a little break, come back at 1:35, where we'll do a Kahoot and also do that extra exam question. So yeah, so I'll see you guys then. Okay, so it's 1:35 again, so we'll continue on. So the next question that we've got is relevant to sort of what we were doing before lunch. So the following statements are about events during an action potential. Potassium, so we have sort of the, you guys can read through this. And we've got the sequence of events here. So you guys can answer in the chat if you'd like, because it won't be a long answer. So which of the events A to F starts the depolarization? So if you just pop a letter in the chat, which of the events A to F starts depolarization? Yep, I've got one answer. Any other thoughts? Okay, so I've got two sort of votes. We've got one vote for C. Got one vote for C and one vote for B. So the answer to this one actually caught me out when I first looked at it. I initially put B, but the answer is in fact C. So the answer is C because depolarization only starts when the voltage gated sodium ion channels open. When the sodium ions are just diffusing in across the membrane, this doesn't necessarily start depolarization. Depolarization is where the membrane potential goes from -55 to +40. And so that is when depolarization starts. It doesn't actually start when the sodium ions are kind of passively diffusing in. So for the next one, which of these events, A to F, requires the hydrolysis of ATP? So if you can now put the correct letter in the chat, and then we'll talk about why that's the answer afterwards. Yeah, we've got one answer. Perfect. So you both got that one right. So we know that ATP is required for active transport. The only stage here that isn't used as active transport is D. So well done, guys. That one was D. Very well done. And that's just what it says there. And then we've got some more questions here as well. Let's see. Got loads of questions. So I just want to ask you, would you guys like to do these questions, more exam questions, or would you like to do a Kahoot? As well, just pop in the chat whether you want to do more exam questions or a Kahoot. Exam questions. Yeah, that's perfect. We'll do some more exam questions, and if we have time, we'll do the Kahoot as well. Yeah, cool. So here's your next question. I don't know how you pronounce that. Synaptophysin. Synaptophysin is a protein involved in the production of synaptic vesicles. Scientists can use the presence or absence of synaptophysin to identify presynaptic and postsynaptic membranes in synapses. Explain why they are able to use synaptophysin for this purpose. So so I'll give you guys a minute just to answer B. Part B of this question. Actually, no, we'll do both B and C at the same time. I'll give you a bit longer. So the first question, we're explaining why you can use synaptophysin. And then the next question is about dopamine. Dopamine is a neurotransmitter. Production of too much dopamine is associated with schizophrenia. A drug used to treat schizophrenia binds to dopamine receptors in synapses. This binding does not lead to the formation of an action potential. Suggests why the drug used to treat schizophrenia is able to bind to the same receptor as dopamine. Okay, so I'm going to give you guys a couple of minutes to answer those questions, and then we'll go through the answers together. Okay, so that's 2 minutes. So let's go through the exam, the answers to these questions. Oh, I'm throwing my pen around, sorry. Right, so where are the answers? Here we go. So explain why they are able to use, so from the question, we know that this protein is involved in the production of synaptic vesicles. So we need to use our knowledge that we've learned about action potentials as to sort of where the synaptic vesicles are. So here's my, oh dear, it's getting worse. Here's my presynaptic neuron, and here's my receptor. We know from what we've just learned about synapses that the synaptic vesicles are found, in the presynaptic neuron. And this is because they contain the neurotransmitters, which then diffuse across the synaptic cleft. So we know that we can use this protein to find out whether we're looking at the presynaptic neuron or the postsynaptic neuron. So that's what the answer is here. Just says that synaptic vesicles are only found in the presynaptic part of the synapse. Okay, so you know that if there's this protein present, then it's got to be the presynaptic part. Yeah, so hopefully that makes sense. Our second question was about dopamine. So suggest why the drug used to treat schizophrenia is able to bind to the same receptor. So you've got to remember that receptors are like quite a specific shape. And so because they're a specific shape, it means that only specific molecules can bind to them. So say, for example, only a molecule in this shape could bind to this receptor. Whereas if I had a receptor that looked a bit like that, you'd need a really funny shape. You'd need a neurotransmitter that looked a bit like that, which would be able to bind to it, sort of thing. So this must mean that dopamine must be a similar shape to this drug, which is used to treat schizophrenia. If it's able to bind to the receptor, then it must be this either the same shape or have a similar shape to dopamine, and it's got to be complementary to the binding site. So by complementary, it just means that sort of these shapes match each other, where one goes in, the other one goes out, sort of thing. So yeah, how did you guys find that question? Was that good? Bad? In the middle? Pretty good? That's good. Well done. Cool. So the next question in this sort of series of questions, dopamine is a neurotransmitter. Production of too much dopamine is associated with schizophrenia. Yeah, oh yeah, we've already read this bit. Suggest why the binding of the drug does not lead to a production of an action potential. So the binding of this drug, which is the same shape as dopamine, doesn't lead to the production of an action potential. So I'm going to give you 2 minutes, because this question is 2 marks, to tell me why binding of this drug does not lead to the production of an action potential. So yeah, write your answers down and we'll discuss them in 2 minutes. Okay, so it's been about 2 minutes. So let's have a little look at the answer. So suggest why the binding of the drug does not lead to the production of an action potential. So although it has the same sort of a very similar shape to dopamine, it isn't actually dopamine. So because these receptors are specific to the neurotransmitter that they bind with, it means that although it can bind to the receptor, it actually can't open the sodium ion channels. And so because these sodium ion channels are not opened, no depolarization takes place. So an action potential is not set off. So if we look at our receptor again, if I draw it over here, so our dopamine has binded to it, not dopamine, our drug has bound to it, but actually the receptor is still closed. That's a really bad treatment. If this is the closed ion channel, and here we have our drug which is bound to it, our ion channel is still closed, and so our sodium can't go through. So our sodium ions cannot get through, and therefore an action potential is not started. And then the other marks you could have got it for, or it opens chloride ion channels, causing hyperpolarization or preventing depolarization. I think these ones are the ones you were probably looking for. So those are the exam questions we've had a go at. So now we're just going to move on to the clinical skills corner, just to finish up for today. So along the same lines, I've sort of tried to relate the clinical skills corner to the content that we've covered today. So I also have one of those here. Got it here with me today. Does anyone know what piece of equipment this is? Can you pop in the chat what you think that might be? I tell you that this end is quite heavy and sturdy. Tendon hammer. Perfect. Yeah, that's exactly what it is. And can you also put in the chat what a tendon hammer is used for? Do you know what it's used to do? Yeah, reflexes. So doctors use tendon hammers to elicit reflexes in people. So so a tendon hammer, so an example of a reflex you might have seen is your patellar reflex. I've got a little diagram of how the reflex works on the next slide. Here we go. So here's your knee, as you can see here. And if the, so here's your knee. And if the doctor hits your patellar tendon with the tendon hammer, that causes a reflex, and it causes your leg to jerk forward like that. Okay. So yeah, and this works by using something called a reflex arc, which is sort of along the similar lines to what we've been learning about today. So the patellar tendon is tapped. When you tap the patellar tendon, the patellar tendon stretches, and this causes receptors in the quadriceps muscle, which are the muscles in your thighs. This causes them to be stimulated. This is sent down a sensory neuron. It's called an afferent neuron in this diagram. Sent down a sensory neuron to the spinal cord, where it goes through a relay neuron, which unhelpfully has been named an interneuron here. And then it goes down a motor neuron, which is here, to our quadriceps and our hamstring muscle. And that then causes our leg to jerk outwards like that. So this is a really clever way of checking that our nervous system is working. And doctors use them all the time. And there are also lots of different reflexes all over our body. So there's not, the patellar tendon one is the kind of the easiest one to do, and it's the most known about, but we've got ones everywhere. You've got them kind of like in your elbow, around on the back of your elbow here, side of your hand, in your ankles. They're everywhere, but it's a good way of checking how your nervous system is doing and what it's working. So does anyone know an example of a disease or a condition that might cause the reflexes to not be working properly or to be a bit damaged or anything like that? Can you pop in the chat an example of a disease that you might know that might cause the that might cause your reflexes to be a bit different? It's all right if you don't know. If you don't know, just put "don't know" in the chat. Don't know. Okay, so examples of these. So it's usually problems with your nervous system. So an example being motor neuron disease. You might have heard of motor neuron disease in the context of Stephen Hawking. This is one of the most famous people who had the condition. But that can cause your reflexes, depending on what kind of motor neuron disease you have, it can cause your reflexes to be too brisk. So instead of your leg just going like that, when you tap it, your leg might go like that kind of thing. Or if you tap the tendon, it might not do anything at all. So that's a really good indicator that something might be going wrong somehow. Other things that could happen. So for example, if you didn't have a patellar tendon reflex at all, you might have damaged your spinal cord. So you've damaged this portion of that reflex arc. And so that means that your reflex doesn't work as it should do as well. So yeah, so so this is really useful. You might have had it done to you at the doctors if they're sort of wanting to make sure that everything's okay. But yeah, so that's just one way in which we use can use our knowledge of the nervous system to look at things like that. So yeah, but thank you very much for coming along to today's session. So I'm just going to pop if you can just bear with me a few seconds, I'm going to put a feedback form in the chat, and it's really useful for me if you guys can fill out those feedback forms. So if you just stay there, I just need to find my email and I'll copy the link into the chat for you. But yeah, honestly, please just tell me if you want more exam questions. Let me know. If you I don't know, want me to go more slowly, literally just let me know. Feedback is the best thing. At the end of the day, these sessions are for you guys. So yeah, I'm just finding the link now. Okay, so if you can just click on that form, and can you just check that it's working for me? But yeah, if you could just fill out that form for me, that would be really useful. But yeah, thank you very much, and hopefully I'll see some of you guys in the chemistry session tomorrow. I'll be teaching that one as well. So yeah, thank you very much.